Hello, my name is Dear Anton Vittorio of CRIM 25, and we are about to discuss the methods of identification other than fingerprinting. Anthropometry, Bertillon system. The science dealing with the measurement of the human body to determine the difference in the individuals. It was introduced by Alphonse Bertillon in 1900s and spread it through the world in 1914. Fingerprints was officially adopted in France, replacing the Bertillon system. Do you familiar in the Will and William West case? In 1903, the Leavenworth, Kansas, when Will West and William West ended up in the jail and had identical Bertillon measurement. Their fingerprints were different, and this started the downfall of this method. Composition of Bertillon system. Anthropometrical signalment, measurement of the body, head, and limbs. Descriptive signalment, a clear and specific system of description of an individual as well as the photography and the male factors. Peculiar mark signalment, by indelible marks, scars, tattooing, birthmarks, moles. Portrait parl, is a verbal and pictures description of a person being identified. Such information may be given by a witness, relatives, or person who is acquainted with the physical features of the person to be identified. Description of the following features considered in portrait parl. Skin color, nose peak, eye color, color of eyebrows, average distance between eye, of eyebrows, eye shape, bird style, chin types, nose, nose length, forehead and cheek, ears, hair, ear, types of lips. Odontology. It is the proper collection, interpretation, evaluation, as well as presentation in court of dental evidence during the conduct of criminal and civil proceedings. It is the application of dentistry in legal proceedings deriving from any evidence that pertains to teeth. Every human body ages in a similar manner. The teeth also follow a semi-standardized pattern. These quantitative measurements help establish relative age of a person. Each human has individual set of teeth, which can be traced back to establish dental records to find missing individuals. Teeth is made of enamel, hardest tissue of the body, so it can withstand trauma, decomposition, heat degradation, water immersion, and desiccation better than other tissue in body. Teeth are source of DNA. Dental pulp or a crushed tooth can provide nuclear or mitochondrial DNA that help to identify a person. Teeth basics. Approximately 30 to 4 types of teeth: molars, premolars, canine, incisors. Teeth differ in size, shape, root, type. Teeth through the years. Childhood, adulthood. Characteristic. Size of tooth, shape of tooth, shape of root, placement of tooth, quantity of teeth, combination of dental, work done: crowns, extraction, bridge, fillings, root canals. Dental work that makes teeth unique: dental crown, dental fillings, dental extraction, dental bridge. Dentures. There are cases where the victim no longer had teeth and wore dentures. Records are also kept for dentures, so this dental appliance is very good source. Bite marks. Impression from teeth found on skin or items left obscene, usually outline teeth placement. Forensic serology. Forensic serology is the application of the study of blood, semen, saliva, and other body fluids. In legal matters, the field generally is comprised of the detection of enzymes and antigens, as in the identification of seminal stains or blood typing, ABO and secretory status, and DNA typing. The serology section of a forensic laboratory may deal with any of all the following: blood typing, characterization of unknown stains, blood splatter, analysis of crime destruction, paternity testing, semen identification in sexual assault cases, DNA typing for identification of victim and suspect. The composition of blood. Blood is a mixture of many components: cells, enzymes, proteins, inorganic substance, salt, water. 55% of blood contained is plasma, mostly water and substance dissolved in it. Most of the solid materials by weight are cells: red blood cells, white blood cells. Forensic characterization of blood stain. 3 questions that must be answered by the forensic investigator. Number 1, it is blood. Use presumptive test. Number 2, if is yes, is it human blood? Number 3, if is yes, can it be associated with an individual DNA? Is it blood? Presumptive test for blood. Presumptive tests rely on hemoglobin's ability to catalyze the oxygen of certain reagents, usually resulting in a color change. Oxidizing agent is usually hydrogen peroxide, H2O2. Visible stains. Castle-Meyer solution for phenolphthalein is applied to suspected blood stain, followed by hydrogen peroxide. A bright pink color indicates the presence of blood. Leucomalasip, green. Same procedure as produce blue-green color in the presence of blood. Invisible stains, luminol, used for large areas where blood is suspected but not visible. If blood is present, it will grow. Must be viewed in darkness and photographed quickly. Red blood cells contain hemoglobin, the high protein responsible for transporting oxygen. Each HB contains for iron containing hems. Luminol reacts with iron. Blood typing. Human serum containing specific antibodies can be purchased. Separately, mix a drop of unknown blood sample with drop of each anti-serum. Characteristic of blood. Class, species, type, disease, Rh factor, antigens. Individual, DNA. Scholar, a class or individual characteristic of blood. Think about it. Blood stain analysis. Categories of blood stain: passive, dripping, transfer, smearing, projected. Projected blood stain occur in shootings, trauma, from blunt weapons, hacking, beating, or slashing attacks.